to Sebastian Dive 712. We are here in Chile off of Chaitén, and you are watching live video footage from ROV Sebastian streaming from research vessel Falcor 2 of the Schmidt Ocean Institute. We're just collecting some Niskin bottle samples here, some water samples. We've just reached the seafloor.
This should be just Niskin 3 or is it 3 and 4?
And then we'll be doing some push cores and some vibrocoring to collect some more sediment here. And I'll hand it over to our chief scientist, Dr. Seb Watt of the University of Birmingham, United Kingdom. Seb, it's all yours.
Okay, thanks John, and thanks for everyone joining us today. Yes, we're on the second dive today, if any of you were also here for this morning's one. As you'll already see, it's looking a bit more turbid, the water here. So earlier today we had some great views of an octopus and a bit of life on the seabed. Not quite sure how much visibility we're going to get here. It's a little bit cloudier. And I think that's probably because we're closer to the river outflow and we're closer to the shoreline. We're pretty close, sort of hugging the coast here, on the north side of the bay that goes towards Chaitén volcano. So we start with a push call, please, Zach, thanks. So we're following the same sequence as we have on previous dives, trying to be consistent with our approach. So we've just taken some water samples and we're now going to take the push cores to get that sea surface material. And the reason we're doing this is we're keen to get that sort of muddy interface right at the seabed. When we start using the longer corer, what we call the vibrocore, to do that, it churns things up potentially at the seabed, and you might lose that uppermost part of the seafloor. And that uppermost part is quite important, both for our biology and chronology. So we want the organisms that are living there right at the seabed today, or rather, in a way, we want to verify that we've captured those living organisms right on the seabed, because that tells us whether we're missing some sediment in our core potentially, and that the sea surface or our core surface is not necessarily the present day. So that's what we're using these push cores for. Visibility is really quite limited, isn't it? You can just see it going down to the sediment there and about to push in. So we'll see how easily this goes in. When we landed the ROV, we kicked up quite a bit of sediment, so I'm expecting it to go in fairly easily. Looks like it is. And in some of these softer sites, if you've seen previous dives, we've had some issues with the sediment sort of dropping back out because it's so soft and loose in this uppermost part. So we'll see. That's a bit hard to see. Looks like we've captured a fairly full push core tube there. So we'll bring that in. So the pilot's just manoeuvring that into the tube holder. Looking like we've got it in with the sediment contained and without disturbance, which is great. We really want to collect these with minimum disturbance, so you can see this is a pretty skilled retrieval procedure to really control these arms carefully, get everything vertical and back down into the holder, and doesn't look like any disturbance at all of this so far. So this will be a useful core, and one in which we can really use to look at that very youngest, most recent sediment and to characterize that. One of the most valuable things we can do between these sites is interpreting one site on its own is quite challenging, but with all these ROV dives and with samples of the seabed in multiple locations now around this system, we can do a comparative study and really start to understand the characteristics of sedimentation at the seafloor, the accumulation rates of that sediment, and essentially building up a spatial picture, a map of sediment distribution and accumulation through this system. And as John mentioned when he introduced, what we're particularly interested in this environment is the impacts and disturbance that was caused by the Chaitén volcanic eruption. Let's let's start with three and see. Yeah. So we'll collect three. Three of these push cores, so we've got plenty of material. And if they all go in fine, so we're collecting this from holder number 3, but this is the second push core. So if these all go in fine, then we can decide if we'd like more material. Just to check, Gianluca, do you want biological material here? Or
No.
you happy not to? Okay, yeah. So I was just saying, yeah, we're interested in the perturbation of this environment from the Chaitén volcanic eruption, which occurred in 2008, so about 16 years ago now. And this was a major sort of environmental event in this region. It was a major societal event as well, so it caused all sorts of impacts in the region, very visible and clear impacts on land, but we don't yet understand the impacts of that event on the seabed, on the marine environment, and indeed the recovery of those environments. And that sort of encapsulates the overall aim of our project here. We can understand those impacts from a geological perspective, from a biological perspective, and indeed just simply in terms of how much material is entering the system and potentially reshaping the seafloor in these environments. We saw that reshaping certainly in the coastal regions with the very rapid growth of deltas at the two river systems that drain the Chaitén volcano. So those two river systems were inundated with volcanic sediment after the eruption, and that really transformed the pathways of that sediment, the channels of those rivers, particularly to the south, to the river that feeds through Chaitén town, which is a small coastal town that was flooded with volcanic material in the aftermath of the eruption. So the entire town was evacuated, had very dramatic consequences for the region. So we got various colleagues from Chile on board with us here. In fact, I think over half the science team are Chilean scientists, which is really great sort of part of this project, really, to be working with such an international and diverse team. I'm going to see if I can get some of those Chileans who have previously worked in the Chaitén region to speak a bit on this dive. They're not in the room right now, but I might see if I can locate them and see if they can come and talk about their experiences, their observations. I think that'd be really interesting to hear from them. So once we progress through the dive, I might see if I can find someone else to come on and talk a bit about that. Because although we've explained on these dives quite a lot about the eruption itself and what we're exploring, there's a few people on the science team that we haven't yet heard from. So I think if we get one more, Zach, please, just a fourth one, yeah, just so we've got a sort of a more full one, or we can sort of choose between them. That would be great. But they all look good so far. The three we've collected. You want a fourth? Yeah. Just so we've got some choice.
Extra.
No problem. So it's pretty soft here, Becky. So we'll see how the Viber call's going.
Yeah.
So yeah.
Yeah, we'll see what it looks like below this.
Yeah.
Well, and then a gravity core.
What's that?
Then a gravity core.
Karis, do you want to do any talking?
Got anything to add?
Okay.
I can do if you want.
No, only if you want it.
Yeah, Karis, come on. Talk to you. I can talk to you. Oh, you did? Oh, good. Oh, that's great. You were right next to me.
Gianluca, do you want to do a bit of talking about what you're doing?
Okay.
Yeah. So sat behind me is Gianluca from the Universidad de Valparaiso, and he is working with another student from the University of Valparaiso, or Universidad de Valparaiso, particularly on the marine biology. So I'm going to just hand over to him to talk a bit about what we've been seeing so far, maybe some of the things we've recovered in previous dives, and what we can see here, although I don't know how much there is that he can talk about what we're seeing right now. But anyway, I'll hand over to him.
Hola a todos. Bueno, ya me presentaron. Mi nombre es Yelcho, estudiante de quinto año de la Universidad de Valparaíso, de la carrera de Biología Marina. Bueno, lo que ustedes están viendo acá, para los que no hablan inglés, estamos trabajando con los punch core. Ya, el punch core es un tubo, un cilindro transparente de un material casi plástico, y la gracia es la recolección de la columna de sedimento sin perturbar, para poder así tener los diversos estratos con la diferencia entre los centímetros que tiene la capa del fondo marino. ¿Ya? Nosotros, como biólogo marino, lo que estamos trabajando es recolectando la macrofauna bentónica, quiere decir todos los invertebrados ya que habitan en el fondo marino, los que se ven así como normalmente, como lo pueden ser los cangrejos, las jaivas, los erizos, las estrellas de mar, pero también estudiamos la infauna marina, quiere decir todos aquellos organismos que estudian, que viven en el fondo marino. Si se fijan en el puchcore, pueden ver como un gusanito moviéndose hacia arriba, tratando de salir a la superficie. Ya, esos gusanos se llaman poliquetos. Ya, los poliquetos, a diferencia de los gusanos, que también son anélidos, o las lombrices, es que tienen la capacidad de poder respirar debajo del agua y tienen una morfología mucho más compleja. Ya hay varios, ahí lo pueden ver, es un gusanito que se ve en la mitad del tubo que se está moviendo. Eso es lo que principalmente estamos recolectando en esta expedición. Ya por las 5 estaciones que nosotros hemos tomado muestras, hemos recolectado harto, ha sido muy abundante, muy rico en especies. Entonces estamos muy conformes con el trabajo que ya se va haciendo. ¿Ya? Nosotros nos estamos muestreando en todas las estaciones porque de repente son muy cerca y entonces la fauna no cambia mucho. Y es por eso que por lo menos en esta estación la biología no la va a trabajar, sino que ya para el día de mañana.
Okay, so we'll try a live record.
So I'm going to do a bit in Spanish and then in English.
So thanks, Gianluca. So talking a bit about the biological work on this expedition and what we've been finding so far. I'm now going to hand over to Javier, who's a Chilean colleague on the expedition, who's currently in New Zealand, and he's here from the University of Auckland. But Javier is a Chilean scientist, and he has worked in the Chaitén region, including right back to, when was your first time in Chaitén, 2009?
2010.
2010. So since 2010, he's been working, studying Chaitén eruption and its aftermath. So I'm going to hand over to him to talk a bit more about that.
Hola, buenas tardes. Mi nombre es Javier Cañete, soy geógrafo de la Universidad de Playa Ancha en Chile, posteriormente magíster de geografía en la Universidad de Auckland, especialista en estudios costeros. Y gracias a la erupción del volcán Chaitén, desafortunadamente tuve la oportunidad de poder realizar mis estudios en el extranjero. En resumidas cuentas, resulta que yo me encontraba de viaje en la Patagonia en el 2010 de vacaciones y solo por curiosidad decidí viajar a Chaitén para ver los efectos de la erupción en la ciudad y en la línea de costa. Resulta que ese viaje terminó cambiando mi vida, determinó lo que pasó después en una serie de eventos, ya que posteriormente a eso obtuve una beca para estudiar en New Zealand, en Nueva Zelanda, y por esas casualidades de la vida andábamos en terreno haciendo un estudio de ríos, un proyecto de ríos con un grupo de colegas, y les mostré información acerca de la erupción del volcán, mi viaje a Chaitén durante las vacaciones, y resulta que a raíz de compartir esa información terminé estudiando los efectos del flujo de sedimentos en la línea de costa. Eso se convirtió en mi tesis de magíster. Luego de eso, en el año 2013, viajé nuevamente a la ciudad, pero esta vez a realizar levantamiento de información, en el cual levantamos información batimétrica, o sea, del fondo marino, pero esa vez solo con un sonar single beam, un monoas que se llaman, que nos permitió capturar parte de la información a baja resolución de los efectos de los sedimentos en el delta, en la zona de la pendiente del delta, y además levantamos información de los sedimentos para poder entender cuál es la distribución del tamaño de los sedimentos dentro del área inmediatamente al frente de la ciudad de Chaitén, además del área del delta que se encuentra adjunto a este en el río Chaitén, que se encuentra inmediatamente al sur, entre medio de Chaitén y el río Yelcho, para aquellos que conocen la zona. Además de eso, a través de la investigación he conseguido adquirir un número bastante grande de información geoespacial, ya que posteriormente o durante el proceso de estudio del volcán Chaitén me especialicé en sistemas de información geográfica. ¿Ya? Trabajo muchísimo con mapas, con mapas de diferentes tipos. ¿Y a qué me refiero con esto? Trabajo con información satelital, trabajo con información batimétrica del fondo marino, y recientemente, bueno, no recientemente, en los últimos 6 años he estado trabajando con vehículos o sensores remotos de pequeño tamaño, que ustedes conocen como drones, pero aplicados a mapeo de alta resolución. ¿Qué más les puedo contar? Bueno, después que terminé de magíster en Nueva Zelanda, el año 2014, volví a vivir a Chile, y el año 2018, con la intención de en algún momento poder publicar los resultados de mi investigación, sobre todo lo que pasó en la línea de costa y cómo la costa ha evolucionado, ha progradado y retrogradado a través del tiempo, volví a Chaitén en el año 2018 por cuenta propia, con una colega. Trajimos en ese momento un dron que recién había adquirido, una estación total GPS, y recopilamos algo así como 1.500 hectáreas de superficie, para así crear una ortofoto, que se llama un ortofoto mosaico, para aquellos que saben un poco, que es como una imagen de Google Earth, pero de alta resolución. En este caso capturamos algo así como 6.000 imágenes desde el dron, y esas imágenes posteriormente las procesamos, creamos este ortofoto mosaico y creamos un modelo 3D nuevo para la ciudad de Chaitén y para el delta. Modelo el cual tiene como objetivo poder analizar la evolución espacio-temporal de la línea de costa nuevamente. Y luego, este año, a partir de este proyecto Fire and Ice, yo me encontraba a puertas de viajar a Chile. Ya sabía que iba a estar en Chile en el mes de abril, y decidí volver nuevamente a Chaitén. Por lo tanto, esta es la segunda vez que me encuentro en el área en un año. Tanto así que en abril de este año volví a Chaitén con un nuevo dron. Tengo mejor equipamiento, por suerte, así que volví con la misma metodología, nuevo dron, mejor sensor, estación total para medir GPS, y realizamos el mismo procedimiento similar a 2018. ¿Qué nos ha permitido esto? Hoy día tengo la suerte de poder decir que tengo uno de los set de datos de mayor resolución para el área de Chaitén, de la ciudad y del delta, específicamente cuando se trata de imágenes aéreas y modelos 3D. Modelos que esperamos combinar con la información que hemos adquirido durante esta expedición, lo cual nos va a permitir en algún momento cercano poder tener un modelo completo del área terrestre, de la zona de la cuenca de Chaitén, la cercanía del volcán, el volcán, y posteriormente la zona de la ciudad y del Fan Delta, además de la parte sumergida. Bueno, eso es todo lo que les puedo contar respecto a mi experiencia, y ahora les voy a dar a aquellos que están escuchando en inglés, les voy a dar un resumen en inglés, ¿ya? Okay, my name is Javier Cañete. I'm a geographer from Chile, but I currently live in New Zealand, and I'm representing the University of Auckland in this expedition. Long story short, in 2010, I happened to be traveling around the region, and out of curiosity, I ended up in Chaitén, seeing the aftermath of the eruption. So randomly in 2013, I was doing my master's degree at the University of Auckland, and I was looking for a research topic. So on a field trip, I share some of my trips with colleagues, and I display information about Chaitén and the time that I visited the area. And they all got shocked and quite interested with the area and the changes that the eruption produced over the coastline specifically. And therefore I decided to conduct my research in the region. So in 2013 as well, came back to the region. I did field work to collect terrestrial sediment samples and to collect single beam bathymetry along the near shore area of the delta, as well as a high resolution DGPS. So, and apart from that, I also did a really in-depth bibliographic revision of all existing data sets, and by that I mean all existing aerial images from previous locally owned authorities. I went to every minister that had relevance in the area that could potentially have information, gathered them all, and I ended up producing a range of data sets that helped me to understand and explain how the shoreline evolved over time from 1998 till 2013, which was the last data set that I had at the time, right? Then after, I moved back to Chile, and due to my former supervisor, Lorna Strachan, who I'm currently working on this project, she happened to be in the US and asked me to produce something extra about Chaitén. And because I love the area and the work that I do, I decided to come back by my own means with a colleague. And by then, UAVs, drones, were around, and they were more available for science. So I had the equipment. I came back with a Phantom 4 Pro and a DGPS station, and I conducted a new aerial survey for the area, a high-resolution aerial survey, and ended up mapping something of 1,500 hectares, which I then turned into an orthophoto mosaic and a 3D point cloud and a new digital elevation model for the city and for the Fan Delta, so I could continue understanding and explaining how the shoreline was evolving over time. It happened that we never got anywhere after I did that site visit in 2018, but last year in 2023, with Lorna, we got back to the site and do something else about it. And the Fire and Ice project was being discussed at the time, and there was an opportunity for me to come back and join the expedition. So, because I was already planning on coming back to Chile before Fire and Ice, this year, in April 2024, I came back to the area, same colleague, new DGPS station, and I brought my new UAV device, my new drones, because I have continued working with UAVs for several purposes in New Zealand. So we came back and we remapped the entire city and Fan Delta at high resolution, even higher resolution this time around, collecting something of 6,000 aerial photos, which then I turned into a orthophoto mosaic again, 3D point cloud and digital elevation model, which are now gonna serve to inform and tell the story of how those changes have been progressing over the last 16 years since the eruption happened. So, which hopefully, well, not hopefully, I believe this all is happening due to the Fire and Ice project, thanks to the Schmidt Ocean Institute. We're gonna be able to produce a high-resolution terrestrial and underwater model combined with all the sediments and sediments distribution around the basin. So we can tell and explain the story of how Chaitén deposits are migrating around the region, and what are the terrestrials and underwater impacts of the eruption. Yeah, and that's me. Thank you.
Okay, thanks very much, Javier. I hope, and it's really great that we have that in Spanish and English, so I hope that's reached some of the people who watch it.
Okay.
But really good to have some insights from Javier, who visited the region several times ago, the eruption and its impacts. So, as you'll see, we've just been talking, we've collected the first part of it, just like an introduction statement. So this one starts here, which is what we've expected from the subvolcanic profile. So this is our sort of geophysical data that lets us visualize the first 10, 20 meters or so in the seabed, particularly in those sort of softer sediment accumulation. And this sort of, hopefully around a 1.5 meter section perhaps that we'll get here, will really give us some insights into the composition of those layers and where those materials can be sourced from. So we're collecting input here through the Chaitén river system. It doesn't look from where we've mapped it that we've got particularly coarse material or sandy material here. But we'll see. We'll get out of course. Sometimes they can be a surprise with the bloody intervals that are visible on the seabed. The core of the last site was actually our longest yet, so 1.5 meters from the first fibre core of the last site, so that gives us quite a long time interval. Of course, we need to work to understand exactly what that time interval is, so we need to use dating methods to get a chronology in it. But 1.5 meters is the longest fibre core we've got yet, so it's exciting to get that. Although when we opened it, it looks fairly featureless initially, but you can see there's layering in there, sort of stretches in there that record specific environmental changes and processes through time, even though things like the colour are not particularly changing. So it looks like the sediment source is not dramatically changing, at least basically colour, but there are grain size changes and potentially other changes that we can look at in detail using mechanical sedimentological methods. So we'll see what we get from this one. But based on the last site, I think we're really sort of working out the variability of this method as well. And we've tweaked a few things with the base of the vibrant cores, where this aperture is what we call the core catcher. And we've adjusted slightly the design of that to try to minimise compression of these soft sediments and to ease the passage of this tube into the seabed. So we're finding in our first few sites we're getting quite a lot of compression, or sort of really strangely in some sites actually limited recovery, which was quite a puzzle because we could see the core tube going into the seabed, and then when it comes back to the surface there was much less sediment in it than we were expecting. It seems this really depends on the sediment properties as well. We've been in quite a variety of sediment types from ground.
If you get it in that white clip there,
So
just
the
that right
Rayas
strip,
River Delta
and then kind of let it fall. It should be pretty close to the end. You can probably catch it.
this site.
I mean,
So obviously the sort of the way those materials
hold your hand there so it can't fall
from
forward.
right core could be quite variable. So we're getting experience So anyway, we'll see what these ones look like. And also learn more about most effective methods to get these various sample types. For the next couple of days, we're going to be approaching the Chaitén Delta. Probably some sites tomorrow where we'll be going right at the front of the delta. And we should really see the influence of the Chaitén eruption in those sediments that we recover there. But or at least that's what I'm expecting to see. This region, it's all slightly complicated because, of course, there's a whole series of other river systems that are entering the marine environment, and those are river systems that were not influenced by the Chaitén volcanic eruption. So we've got a mix of sediment sources that are sort of competing with each other through time. And the volcanic eruption marks a transient event, a short-lived but extreme disturbance that is sort of imprinted on the background that we'd expect to be more continuous and less variable. So where Chaitén town is situated, as I've said earlier, it's on the Blanco River, which is this fairly small river system that drains Chaitén volcano. But Chaitén also sits just north of the mouth of a much, much larger river called the Yelcho River, which has a drainage basin extending over a much larger part of this region. Now, the Yelcho drainage basin was not strongly influenced by the Chaitén eruption, but in the longer term, it delivers a lot more sediment to the ocean than the Blanco River. So, what we're seeing now is likely a mix of those influences, and we're targeting dive sites to try to sort of select regions that we think will
do another one now, so we're
yes,
gonna move
please,
a little
another
bit,
one.
right?
So just to move a little bit,
Right,
please.
I'm gonna
Yeah.
come up about 20 meters so we can just make sure we can see that that's in there, right?
Yeah.
And then we'll
Okay.
land again, because I gotta move anyway, right?
Yeah,
So
yeah,
at least
makes
we might as
sense.
well make sure we're in there, right?
Perfect.
Let me get.
Okay, thank you. So yeah, so this site we'd expect to be more influenced by the Chaitén River, but we'll learn more when we're able to open one of these cores on board. And as I say, tomorrow we're going to aim for sites that we think will be influenced separately by the Blanco River and the Yelcho River. So the reason we're getting two on these dives is that we have a duplicate. We can't do everything that we would like to do with these sediment cores on the ship. We've got a pretty good setup in the lab, I have to say. So we've various scientists have equipment on, and we're sort of spread right through the lab. So we've got the biologists with their stations to be looking at the sort of microscopic organisms and particularly the shells of those organisms that are preserved in these sediments. And then we've got the geologists who are studying the sediment grains, particularly those from terrestrial sources, so they're looking for volcanic grains. And we've got a setup as well that allows us to photograph these cores, and we've been trying to also get some chemical information from scanning them, and even some magnetic information. So there's a whole range of methods that we're sort of trying on these cores, but it's not the setup that in a full sort of core scanning lab that you could do. So we're opening one core, getting a lot of information for our initial descriptions and photographs and what we call stratigraphic logs, so detailed descriptions down the core sequence. But we're leaving the longer core and what we think is the better preserved core on each dive. So we're sort of making a selection when we recover these. We're leaving that one sealed. So that's sort of archived for now until we can take them off the ship, return them actually to Santiago, to the Universidad de Chile, and then thereafter they will be worked on in a sort of more controlled environment. So we'll initially do some scanning through the tube before they're opened, and then they'll be split and further scanning will take place, and then they'll be sampled. So all the samples we're getting now allow us to understand what we're recovering, to do some initial analysis, and then we can build on that later after this expedition. So we've come up off the seabed because the visibility here is really presenting a challenge, and one of the things that's always important to check is that we've got the core back in its holder properly. We don't want to risk losing it on the ascent. We don't want to risk sediment falling out of the base of it. So we've come up a bit to get a clearer view because it's just so turbid here, there's so limited visibility. And then we're essentially going to go back down, having moved a few meters, 2 to 5 meters or so, just so we're in a cleaner side to collect the second duplicate vibrant core. So currently about 23 meters off the seabed. Still quite a bit of sediment around, as you can see, obscuring the view. Not a lot for our biological loggers to be logging right now. Have you seen anything interesting on this dive so far? No. No, with the limited visibility, it's pretty hard to see things here.
We can barely see the vibrant cores.
You happy that one's in the holder, Zach? Yep.
Happy I'm gonna get. Working in the fog.
Okay. It's looking good. So you're gonna get the second one in the holder while we're up here?
No.
No? You're gonna wait till we're down?
Because then if we drop it while we're up here.
Ah, I see, yes. You're never gonna find it. Yeah. Makes sense.
So I'm gonna land again.
So just getting into position to get that second one out, but we're going to go down, as H was just saying, before we start moving this core tube around, because if it gets dropped here, it's going to be near impossible to find in that soft mud at the bottom.
Yeah, we definitely won't be able to see it.
So I can just about see the number 3 written on that one. Or is that the markers as well? Yeah. See, we've got some depth markers on these, which helps us understand how far we're getting in as it goes down. Although with the visibility here, that's pretty challenging to see those.
Did he come on the bottom?
Yeah, I think we must be with all that mud kicked up.
Couldn't even see it come up this time.
No, I was clearing a little bit below. Oh no, that's just the top of the bush, guys.
And the currents aren't too crazy right now?
Almost none. I wish there was more.
It's weird.
So they went from too much to none.
Yeah, so just talking about the currents, and the currents have been causing quite a lot of challenges at some of these sites, particularly offshore the Rayas Delta, which is another river system draining Chaitén that we were sampling a couple of days ago. But this river system's sort of in quite an enclosed bay, and we went from sort of extreme currents the other day, which really sort of limited when we could perform these dives. So we were having to work very carefully around the tidal cycles and find that period around high or low tide when the currents were at a minimum, because they were switching direction, and having to do pretty fast dives because of that. And suddenly, just around the coast, I mean, we're only a few miles away, 10 kilometers perhaps. The currents here are sort of down close to zero, which is partly what's limiting the visibility here, so nothing's being swept away. And this is what we've been picking up on sort of a local scale, real variability in these currents, and that's having a huge influence on sediment transport. We can see that in our sub-bottom geophysical profiles when we're imaging the seabed. You can go from an erosional region where you've got sort of a bare seafloor exposing pretty old surfaces, to a depositional region where we're building up fine sediment in a very small distance. Partly this is due to quite complex currents, I think, in the region, partly influenced by these river systems, but also influenced by the underlying topography. The seabed in this region is actually, although it's quite shallow, 100 to 200 meters, it's very complex in its form. And that's predominantly linked, well, it's linked to two things, I guess. I think it's linked to the tectonics of the region, So there's a lot of crustal faulting. We're near a plate boundary on a subduction zone, and there's a major fault system called the Liquiñe-Ofqui fault zone, which is not a simple fault structure. It's not just one fault. It's a series of sort of splayed faults coming off a main fault zone, and those faults sort of crisscross the region. So first of all, those cause sort of patterns of erosion and topography that have also then been shaped by glaciation. So this area was also covered by ice sheets at the last glacial maximum. So if you were here 20,000 years ago, this would have been covered by thick ice, several hundred meters of ice. And that ice has also sculpted the seabed. So the seabed is marked by sort of complex channel-like features, highs. These aren't sort of simple sort of down gradient channels like sort of river channels. You know, there's a range of sort of isolated deeper areas, shallower areas, and this is causing really real complexity in the ocean currents, the mixing of water bodies right through these inner seas of Chile, and that's really complicating the sediment transport pattern. And really, this is the first time we've had sort of detailed high-resolution data through which we can explore this. So we're learning a lot about the currents, about how that's linking to the sedimentology, about how that's dispersing these volcanic sediments, and indeed, therefore, the consequences of those volcanic sediments for life in the region and for the marine environments. I have to say it's a more complex picture than I'd envisaged, really. So, you know, there's a lot of interesting things that we're learning. And it's quite exciting to be sort of getting a clearer picture of all those complexities now we have these higher resolution data sets. But lots for us to be working on during expedition and indeed for some time afterwards, I think. And that'll rely on the cross-disciplinary team that we have on this project. So the oceanographers, the marine geologists and micropaleontologists, the marine biologists. Oh, there we are. That's Becky. Becky's coming into the room, whooping at the mention of micropaleontology.
Yes,
So-
we have some. We also have a micropaleontologist in the room. Jacob held
So we have Jacob sitting. So that's Jacob is a master's student with Becky. Is that
Master's
right?
student,
At the
just started.
University of Alabama.
Roll Tide.
He's from Alabama.
You know,
And
we
then-
even had someone on the chat saying that.
Oh, really?
So you're obviously yeah. I mean, maybe they're from Alabama. I don't know, but.
And then we've got Amalia from Universidad de Chile, and she's been so inspired. She's been drawing forams in her notebook today since we saw them in the scope
Really?
yesterday.
I just
Beautiful. She's a great artist. Sorry. I
So yeah, no. All I was saying is, you know, it's a team like this. You know, and that's a really exciting thing about this sort of project. You work with people from all sorts of disciplinary backgrounds. So my background is volcanology. We've got people here who are sort of more sedimentologists, people who describe themselves as geographers, like Javier, glaciologists, micropaleontologists. So a lot of people who are broadly geologists, but from quite different disciplines within geology. And then a team of geophysicists doing this marine mapping, which is really key to all this work as well. You know, when I work on land field projects, I don't think I ever work with such a diverse team of scientists. I wouldn't do those projects with biologists, for example. And that makes marine science really exciting and sort of enriches it, I think, in terms of the range of questions we can address. And actually, you need all that input to fully interpret and understand the data sets that you're collecting with marine science. I think you're seeing even less than we were before, Zach.
Yeah, I
Can
think
you-
probably whatever we kicked up is just
Yeah.
no current to take it away, unfortunately.
Can you see enough to even get it in?
Yeah, I think we can do a vibe report. I'm not sure we're gonna be able to scoop it. We can't see the
No,
current.
that's fine. We weren't. The scoop wasn't planned, so that's fine.
I think we can probably vibe report.
But yeah, we
Yeah,
literally
that's
can't
good.
see the seabed, so.
Yeah. That's fine.
So I think we'll try the second vibracore, and then I think that'll be it for this dive. So quite a short one. But first of all, let's see if we can get this one in.
Yeah, so far you can see enough to see the top at least. So if I can see the top, I know I can
Sure.
move it, you know?
Yeah.
Joel's using his Jedi Force.
He does look like a Jedi.
He's doing his best imitation of Ben. But I just think Ben's doing this all the time.
Yeah.
Well, at least Ben's a fairly older gentleman who works here.
He's the Alec Guinness of this operation. Very cool.
Yep, you can go ahead and let him. Got your view on the minute cam there too, if that helps.
Yeah, that's right. The pilots are certainly making quick work of this, as someone's just said on the chat. We're getting very smooth operations just a few days into this expedition, you know, really getting quick at collecting these push cores, transferring the vibracores, and we're getting some really good cores in now, as I was saying earlier.
It kind of looks like the current's going the other way now, like going from behind me to forward. It's just messing with us. I am happy to release and let it drop a little bit.
Question about micropaleontology, or well, a comment really about micropaleo. I don't know if you want to come and talk about imaging these and how big these things you are, so that you're studying.
I
So,
am ready to
but-
go in if
Jacob,
you're
do
ready.
you want to chat?
yeah, why not, Jacob? Do you want to come and talk about diatoms, or so we don't need an SEM. Someone has on the chat has said, do we need an SEM to see these? We've been looking at these under an optical microscope, but we've certainly got a high magnification microscope here to look at particularly the diatoms, which are very small indeed. I am not the person to talk about how small they are. That's something that Becky can talk about. So I'm just going to hand over to Becky.
All right, and the question is about, let's see this. Oh, it's mainly about how challenging micropaleo is. Yeah, big respect to anyone who can sort out those little bugs. Yeah, so micropaleo is certainly one of the most time-consuming activities of what we do here on board. We have all of our students and postdocs and PIs contributing to making slides of the sediment that we're finding in the cores, as well as sieving through the sediment, washing out the mud so that we can look for tiny shells of microorganisms with special interest in those called foraminifera. They're about sand size, so we wash them with a mesh that's very fine. It's 63 microns. It takes a long time to sieve these, and Amalia's kind of like giving a blank stare off into space right now because she's been doing a lot of sieving, but really, really good at sieving. Jacob's been sieving a lot. He was up until midnight last night sieving. So we're washing out the mud to get all the sand-sized particles, and then what we're going to do, and this is something that the reason Jacob's here is because he's been picking these forams for the last 2 years in my lab, working on forams offshore Mississippi. But of course, you know, forams are useful in so many different environments, really only in marine and brackish settings, but they're very useful for learning the details about the water temperature, salinity, bottom conditions. So we're doing 2 things on this cruise, which is washing the sediment for those microfossils, for the foraminifera, to get information about their populations and how they change through time. That is, how they change with depth in the sediment record. But we're also staining the very top sediments. So when we bring these up, we're making all these notes about the push cores and the vibracores and how well we recovered that sediment, because we're going to take samples of it, of the very top of that sediment water interface, that very top of the seabed, and we're going to then stain them with something called rose bengal that only stains living tissue. So if we are able to take that uppermost sample of the seabed and see what's living there today, as you can probably see on your screen, we've got all the context of water temperature, salinity, oxygenation, and we can visually see how murky or turbid the water is, what else is living there in terms of macrobenthos that the biologists, the other biologists on our team are interested in. Our friends from Valparaiso who've been looking at we got some thumbs up here, that are looking at things like worms and echinoderms, like sea stars and all kinds of things. So we're combining all of this information in the context of what's living there today to best understand what these different species prefer, so that then when we look in the past, when we look into the sediment record and look at older and older and older layers, we can use that context of different species, what prefers to live in different environments, to then reconstruct past environments. And of course, the most important environmental change that we're interested in are the changes that we see from before eruptive events and then after eruptive events. So when we have a disturbance in the system, like a major volcanic eruption, that would be represented in the sediment record with something like an ash layer, which we've seen in cores already on our expedition here in southern Chile. When we see that disruptive event, we can look and we can sample the sediment just below to get background conditions, and then just above, and even take several samples above in sequence to see exactly how the marine communities, these organisms, then responded to the event and recovered from the event. And why is this important? So it might just seem like a fun exercise to see how the marine communities change, but it's actually got some human impacts here, because there are a lot of communities that live in this area that rely upon seafood. They rely upon the health of this ocean to live. Like, that's their livelihood. So a lot of what we've seen and a lot of what we've had to navigate around as we're operating here on the Falcor 2 is salmon farms. There's a lot of fishing that's going on here, and a lot of very sensitive ecosystems in these marine basins that we would want to know, how healthy is it, and how does that change with eruptive events and also with human impacts. So there's a lot that we're doing here that can help inform the sort of fisheries and aquaculture industry of southern Chile, to help with those local communities that really affects the entire country. And really affects everyone, right? A lot of our food does come from Chile. It's a very productive agricultural region. So, yeah, that's micropaleo in a nutshell and what we can learn from it. I could keep talking, though. I only talked about forams. If I put Jacob on, we have said he's a diatom whisperer.
Diatoms, yeah.
Do you want to talk about diatoms?
Discussion. I was all there waiting for diatoms.
Oh gosh, well, I think Jacob's the best person. I'm putting him on the spot here. He's a new graduate student. He just presented this, actually. He's taking a class as a brand new graduate student. It's called Climate of the Common Era. And here we are, trying to understand ocean basin changes, volcanic changes, environmental changes. We are doing Climate of the Common Era right here as we speak. So he and our PhD student Mane just presented on what they're doing here. And so Jacob, I think it's time. Want to talk about diatoms for a minute?
I can talk about diatoms.
All right. He loves diatoms.
I do love diatoms.
This is why he came to grad school, because he just fell in love with diatoms.
Yeah, so diatoms are these amazing cellular species, protists to be more exact, that are very, very beautiful. They have immaculate silica glass houses that encase the main cellular components that make up the cell. And with that comes a lot of, like, for instance, they actually make up about 40% of our atmospheric oxygen. And as far as how difficult it is counting them, looking at them, taking photos of them, it can get pretty strenuous. Staying at a microscope for 4 hours, 6 hours potentially, sometimes, it gets pretty exhausting, but it is so fun to look at them. You have to take a look at some of the species. I mostly look at species from the Antarctic Peninsula area and studying glacial-marine diatoms, and they are very pretty. And especially in the Antarctic, diatoms tell us a lot about seasonal change in the atmosphere and in the climate of the Southern Ocean. And we're doing something very similar here, looking at those seasonal variations, and I plan to hopefully use this as a comparison. And it's good practice to look at diatom change, even if they're different populations, diatom change in seasonal variation. Because with seasonal variation, especially in Antarctica, you have meltwater that can bring in a lot of iron, and diatoms in particular, they use iron as a main source. Most of us have learned in elementary school or middle school, high school, the photosystems. And the main component of those photosystems is P680. And that includes that iron. And in order for photosynthesis to occur, that iron has to be there. And especially in Antarctica, and even in this area where we're seeing ash deposits that contain iron, we can see, hopefully, we'll be able to see some sort of diversity increase, population increase, seeing this iron input affecting the diatom populations. Because if there's more iron, then there's either more photosynthesis or the diatoms get larger. It depends on other variables, including salinity, temperature, depending on maybe they get larger as the nutrients are put in. So diatoms are very, very fun to look at, even though it takes many hours to count them. I myself have counted probably well over
15,000.
probably 15,000, yeah. 15,000 diatoms. I'm very familiar with these guys, and they tell us a lot more than you may think, even though they're so small. So I'm giving it back to Becky.
Thanks, Jacob. That was awesome. And we could just hear your passion for diatoms come through. Claps. So it's very rare to actually have an expertise in both two different types of microfossils. And Jacob here has worked for almost 2 years on foraminifera in the Gulf of Mexico. And then he started working on diatoms, and that's when he went, I have to go to grad school. I have to do this. So now he's starting his master's. He just graduated. But those are two different types of microfossils. In the chat, someone mentioned nanofossils.
Ooh.
So nanofossils are even smaller.
Only if we had Will.
Yeah, and we actually have another student at Alabama who studies nanofossils. He'll be graduating soon. But there's a lot we can learn from all the different types of critters in the ocean, from the smallest scale to the largest scale. And nanofossils are certainly the smallest scale, and it takes real expertise. There's maybe only 20 people in the world who know nanofossil biostratigraphy, just how they change with the layers in. And we can actually use these different fossils to tell time, actually, when we go really far back in time. But nanofossils here, like in today's oceans and most likely here in southern Chile, they're not really diverse. The nanofossils that we would be referring to are called coccolithophores. And yes, they're tiny and they're also very beautiful. But we don't have a lot of diversity in the modern oceans. Mostly what we see is this what my student Will calls the cockroach of the modern oceans, because they're everywhere. Emiliana huxleyi. You can look that up if you guys want to go deep, go small. But yeah, Emiliana huxleyi is probably mostly what we'd see around here. And it wouldn't tell us as much about environments. So that's why we're so focused on the forams and the diatoms. Of course, I say this not being a nanofossil expert. Somebody gets on and wants to share a little bit more about nanos, please do. But yeah, here we're going to be looking for forams and diatoms, which is also very tedious work, but we should learn a lot from it. Yeah. Zeb?
That's great. Thank you. Thanks, Jacob. Really good to hear from you, and good to be learning about everything you're working on. And as Becky and Jacob have been saying, micropaleontology is really key to so many fields to reconstructing our long-term understanding of climate and environment. You see those records of past climates, of past processes reconstructed from the geological record. Key part of that data will all be derived from these sorts of methods they've just been discussing. So these tiny fossils hold really key information, important information for us to understand Earth systems. And that's exactly one of the methods we'll be using on these cores that we're recovering right now. So you see this is just being put into the holder. The visibility causes real challenges on this dive. Difficulties getting it into that sort of black casing at the base. I think still not quite got the bottom part in. It's looking. It's very hard to judge, actually, as you can see. When you're just doing this with the camera images, it can be quite deceptive. To get the alignment right, to get it into these holders.
You might have to push down to where you can slip your grip down. You know what I mean? If you got too high of a grip now. You know what I mean?
So we're currently about 20 meters off the seafloor here. That's about 80 meters below the sea surface in this part of southern Chile. And we're really close to land here, so can't see anything right here from the control room of the ROV. But if I go upstairs in the ship, we'll get a view of forested slopes. Really dramatic topography, actually. We've had a lot of cloud. Pretty much since we left Puerto Montt at the start of this cruise. In fact, I would say today's the first day we've had anything like clear skies. It's not been clear, but we've seen some blue, and that's given us some spectacular views of the town of Chaitén and the landscape behind. So as I say, the cloud hasn't completely lifted, but we've been able to see the snow-capped peaks around Chaitén, and it's a landscape with really dramatic topography, very, very steep slopes, sort of fjord land, and that reflects how it's been shaped by glaciation in its recent history. And it makes it very visually clear how this sort of sediment that was blanketing these very, very steep slopes would have been so easily remobilized by rainfall after the volcanic eruption of Chaitén in 2008. So it was covering slopes where it was never going to be stored long term, and this just meant it was very, very quickly retransported as soon as there was heavy rainfall after the eruption into these short, steep valleys, plunging down these valleys in fast-moving flows that we call lahars, which are sediment-laden flows, volcanic mud flows. And essentially these short, steep river systems funneled all this material down, and they slow down when they reach lower gradients. And Chaitén town sits at the foot of one of these valleys, so essentially that flow slowed down when it reached the coastline adjacent to Chaitén town, and at that point that river channel could no longer hold that flow, so that all that sediment being carried by this lahar, by this volcanic mud flow, flooded over the banks of the channel. In fact, it to the sea through the central part of the town, and that is now the modern river channel. So the whole form of the river system changed, cutting a new channel, a shorter channel, and building a delta at the front of this channel. And where it spilled over the banks of this new channel, it flooded through the town, leaving behind deposits a metre and a half or so thick of volcanic sediment that was left behind when the water drained away. So a really dramatic change and impacts on this town. The town had been entirely evacuated in the days before this event. I mean, really within a day or two of the eruption, the town had been evacuated. Had to be very rapid decision making around that, but recognising the risk of these lahars. And we had a seminar from two of our colleagues from Sediment, the Chilean Geological Survey, a couple of days or so ago on the ship. And these are scientists whose job is directly to work in the hazards team at Sediment, the hazards and risk team, assessing volcanic hazards, landslide hazards, and so on. It's really interesting to hear from them and their perspective around. Excellent, well done. So you put it back in the vibracore holder. Yeah. Okay. So really interesting to hear from them about their experience and their work, and knowledge of the region, of hazards within the region, and to be discussing some of the challenges around decision making around an event such as the Chaitén eruption. So if you've been watching the video, you'll have seen that second vibracore, we were really struggling to get it into the holder with the visibility, and then now being off seafloor. So it's gone back into position on the vibracore for our ascent back to the surface. Yeah, we got two. Yeah. They both went in, yeah. The barrel came off the top. They both went similar depth in. So you'll see it seems to be very soft sediment.
Okay.
Soft, so yeah, it's
So hopefully
interesting.
we'll have to.
Well, hopefully, yeah. So now position, I wasn't expecting to be quite so far.
Here,
Roger that. We're just staying where we are until you say so.
What?
Yeah.
Alright, come check.
Touch left, clear, buddy.
Hey, Seb.
Hey, Sophia.
Thanks so much for narrating. Our floats are on surface, so would you like me to give an outro, or would you like to give the outro?
I'm happy for you too, Sophia, unless there's
Okay,
anything you want me to
sounds
say.
good.
But yeah, so yeah, as you say, we're just coming back to the surface, aren't we? So how many meters have we got to go?
90.
90 meters to go. Yeah, okay. Yeah, happy for you too, Sophia.
All right. So I think you probably hear in the background some of our ClearComm communications going off. That's because a lot of our pilots have exited and are waiting in the hangar to retrieve ROV Sebastian. So we're on our way up. It's going to be a super short ascent of about 90 meters, as I've said. Thank you all so much for joining today. It was a bit of a short one as we collected some more vibrocore samples. It's going to be excitement back on deck as our team is processing them. And as always, stay tuned for our next dive, and don't forget to be subscribed to Schmidt Ocean. We're also on various forms of social media if you want to follow along closely what's happening this expedition. I hear maybe soon we'll publish our first expedition video where we're going to explain exactly what we're doing. Made by our very lovely multimedia correspondent, Alex Engel.
Copy that. We're passing you control.
And so with that said, thank you so much for joining us, and I'll see you all next time. Take care.
Looks like I got it.